just a little bit of recap from the last time. So, if you open up your history, the history scientific argument writing module in SLS, if you recall that. So the last time we looked at a few essay samples and we assessed the essay samples using this rubric here. Okay, but today we will try to do a bit of an outline of some of the essays. Okay, but we'll do it step by step. Okay, so the main thing in the rubrics that I think that students struggle with is the first one, argumentation, and under the content it has a comprehensive selection of facts. Okay, so these are the two things that we are going to focus on. I will do the content part first because I think it's more straightforward. Then we will do the argumentation after that. Okay, so if you look at part 4, activity 4, that's the content part. So for content, for history level for content, you need the depth, but at the same time you need the breadth. And I would say they're equally important. Okay, so for the breadth, your learning, your rubric says that you need to have a comprehensive selection of facts and principles that is relevant to the essay. So what does comprehensive mean? Graph number is at least 7 topic areas. Okay, the topic areas depends on what questions are looking for, which you will see later on. Okay, so for example, this first question, right, is how the diversity of rows of proteins that are relevant to living organisms. So the topic area is based on the topic of the question, which is the diversity of rows of proteins. So you need to give me different types of rows of proteins, and you must have at least 7 different types. They must be from different living organisms. Basically, that's the first question. Okay, so you need to consider how many types of figures. Don't just list down all the rows, but you must make sure that you cover sufficient breadth in terms of different areas and in terms of the contextual living organisms. So you must have different living organisms. Okay, so that's the last one here, which is the content and context must be addressed completely. So content being the rows of proteins and the context in this case the living organisms. Okay, so we'll do a few practices. There are 4 questions here. There are 5 questions. I don't mind. Okay. Each of you do 2 questions. So maybe Arthur and Shao and Shengge can do the first question. Then Shao and the 3 of you can do the second question. Then the third question, the 2 of you. The fourth question, the 3 of you. Okay? So you list down the topic areas that you can think about for the question that you answer. I don't need you to give me all the content, just the topic you will. How about you? How about the content? Especially relevant figure out the content. Okay, so let's do each question one by one, okay? So the first question, discuss the diversity of roles of proteins that are relevant to living organisms. So you need to have diversity of roles, and that is how you define your topic area. So the different topic areas will be different roles of proteins, but these different roles of proteins must be from living organisms. So you must exclude prions, you must exclude viruses. Okay. So if you were to look at this list here, so you have cell signaling. Yes, that's one category. Okay, so you have cell signaling. Then you have notice in the list of examples of other cell signaling that they put in receptor proteins in cell signaling, kinases, phosphatases also cell signaling. But antigen antibodies, while it is true that it will eventually lead to some form of cell signaling, but the main function which they are in is actually under immune system. So they can be under a separate category. Okay, so they can be under another category on immune system. Okay, so that will be your antigens, antibodies. You all will learn more proteins when you go into the immune system. Okay, but anyway, the next one, DNA polymerase, other enzymes, ribozymes. Here is the part that is a little bit weird. Okay, I will ignore the ribozymes because that's RNA and that's not protein. Okay, but if you were to look at DNA polymerase and other enzymes, they are all under the function of, all under the category of enzymes. So they are basically just enzymes. Okay, so enzymes is another category, but everything and anything that is enzyme related is all under enzymes. Okay, that includes what you write under photosynthesis when you write about Rubisco, ADP synthase, all these things are enzymes also. Okay, so it will be under the topic area of enzymes, it won't be under central dogma. So don't categorize your topic area by the topics that you all learn in H2 or H3. You need to categorize it based on what the question is asking. Okay, so that's one. But if you were to bring in silencer is again not a protein. What's the protein? Yes, it's repressor. Okay, but that is on regulation of gene expression. Okay, so that's another category. You can bring in repressors, you can bring in activators as well. There are other proteins also. Okay. Initiation factors, etc. So don't have to restrict to transcription, can also be translation fine. Okay, so that's regulation of gene expression. Then you have transport, okay, transport across membrane. So you have all the different channels, carriers, etc. Transmission channel, that is also a channel. Okay, homostasis when you involve hormones. Hormones is also part of cell signaling, so I will actually put them together. Okay, and then there is photosynthesis. Photosynthesis, besides those enzymes, what else do you know? What other groups do you know? Enzymes, what are proteins? Electron transport chain, which is linked to photosynthesis and also respiration. Carboxyl example, collagen. Another one hemoglobin, again, hemoglobin is a transport protein. So you can have transport across membrane, you can also have transport in circulatory system. Okay, and the last one, enzyme reaction. Okay, so from here you have 1, 2, 3, 4, 5, 6, 7, 8, 3 more than enough. Okay, so you just need to pick 7 of them, at least 7 of them, good to have an extra one, and then you elaborate for each one, talk about the examples that you know of, what kind of living organisms have those proteins. It could be all living organisms, it could be specifically plants, photosynthesis, or certain types of plants, for example. Okay. Then give a few examples as well, and that will be everything. Okay. But that's the content part of it. We talk about the argumentation later on. Okay, so this is the first one. Second one is flows of ions in living organisms. Give me the question very carefully. Flows of ions. It's flows of ions, not ionic bonds. Flows of ions, not ionic bonds. Okay? Similarly, not transport of ions, it's flows of ions. So membrane transport of ions involves the membrane transport. So, you're transporting the ions, but it's not the function of the ion, right? But the channel is a gradient. Ion gradient. You can talk about ion gradient as a category. So ion gradient is needed for what kind of function? Right? Generation of action potential.
The proton gradient for respiration and photosynthesis.
The proton gradient. For synthesis. In respiration and photosynthesis. Okay, so that is ion gradient. Then you have nervous, okay, but this is under nervous system, okay? So you can separate these two as 1 and 2 because they are after all different functions of gradient. Active transport is a mode of transport, it's not a function of ion. So what is that ion that does transport? Yeah. Okay, so when you talk about action potential, you need to bring in the different ions, your Na+, your K+, they are part of it. Okay. Then proton gradient is your H+, that is a function. So it's your H+, this is your Na+, K+. If you want to bring in the other nervous system stuff, you can talk about Ca2+ as well. But that's more for the synapse part. Okay, then proteins. Some channels need ions. Say you give example of a channel that is that. What does it mean? Okay. This is more of gated channels. Gated channels. Okay, so ligand-gated channels requires that ligand, which the ligand can be ions, which means that you are saying that the function of the ion is as ligand. So this can be another category, ligands. So this is the third category. Not so much the channels, but them as ligands. Okay. Which is kind of part of this, but it's part of our synapse stuff. I cannot remember about it. Then you have proteins. Proteins are group interaction, as I mentioned. The question asked of ions, not for ionic bonds. So our interaction is not
ions that form negative create like for example the ion is the active side, right? Then the ions can attract also.
So I was thinking more of like those aspects of the specific proteins that give the protein this shape and therefore its function. Then without these ions,
the R group of the, is that considered an ion?
Alright.
The R group is
Has
I don't really consider that as ions though.
that has a charge.
Do you consider R group that has a charge in the protein? You have a long protein sequence, then the R group, is that an ion? It's not really an ion. I don't see it as an ion. Ion I see as a small molecule with a charge.
Amino acid, a single amino acid with its whatever.
The filter ion, they can count as ion.
That's an ion. But on big
On a big protein,
polypeptide,
I wouldn't consider right.
there are charge areas, but I'm not sure whether it's good to call that an ion.
Yes, correct, that's it. You think? Yes, you can talk about filter ions. So you can use filter ions. Yes, pH buffer. So the function is more of pH buffer, so that's the fourth category. Okay, enzymes. Actually, for static group, we can bring in group. Okay. Enzymes. What about enzymes? Okay, so those will be more of your cofactors. For example, Mg plus in your RNA polymerase. Okay, what else do we have? Photosynthesis respiration, where's the ion? Proton gradient. Phosphate backbone, again, I won't really consider that as ions. But if you want to count the ion, you can say phosphates are ions. I'm going to give phosphate backbone, but I want to talk about self-signaling. Phosphorylation cascade, self-signaling. So 7 areas. So be very careful in terms of how you define the area. You must always go back to the question to make sure that it is really addressing the topic in the subject of the question. Okay. And again, living organisms, right? So there must be different living organisms here. What I'm seeing here, a lot of animal stuff. There is no plants. You need the diversity for this. I think this will plants. But any other things in plants? Sorry? Oh, transport. Cofactors, yeah. Cofactor, what is that part of coenzyme? Coenzyme is not. Coenzymes are organic. Yeah, but we can talk about the chlorophyll. Chlorophyll has an energy charge in the middle. It's very similar to heme, but yeah, it's also a prostatic group. So this is prostatic group. Sorry, very messy. So this is another example. Okay, that's about it. Okay, so that's question, the second question. Third question. So these two, the first two is about rows, right? The next two is about comparison. Comparison is a bit more tricky in terms of how you identify the topic area. So describe the structure and function of ribozymes, and then the second part, compare this with the structure and function of enzymes. Okay, so you have structure and function of ribozymes. In fact, I will actually put one area as structure of ribozymes. So this is one area. Then another area is functions of ribozymes. Okay, requires you to talk about similarities and differences. So you need to have similarities, you need to have differences. So under similarities, you can compare similarities in the structure, similarities in function. This will be different areas as well. Similarly, for differences, you have differences for structure as well as for function. So in this case, I can only think of 6. And it's okay to have just 6. Okay? The 7 is just a rough number. Sometimes it's only 6. Just that you need to elaborate more for each one.
Similarities, they will have primary, secondary, and tertiary.
Yeah, okay, I don't care the content. Okay? You can only think of the content yourself, but compare yourself. Basically, these are the areas, and you need to have points for each area and elaborate each area. Okay? I think we all know and forget all about ribozymes already. Okay? So you can revise your ribozymes and then you can answer this. Okay, let's move on. The last one. Okay, so the last one, compare polymerase structure and binding sites of hemoglobin and immunoglobulin IgG. Explain why IgG can bind to a greater diversity of molecules than hemoglobin. So you have a part that's on compare, and then you have a part that requires you to explain.
Wait, sorry, can I,
Yes.
I forgot to add it, because for my explanation, I only added topics, I haven't added like structures, comparative structure.
So you want to add what you want to add?
And it's numbers.
Or you tell me I right. So comparison, you have similarities and differences, right?
Yes.
And then, so you need to compare similarities in terms of the quaternary structure and similarities in terms of binding sites. And then you need to compare differences in terms of quaternary structure and also binding sites. Okay, so that's 4 areas. Then what else?
And diversity.
Then it's on diversity. So how do you speed up to have diversity? So how do you have the other areas here? Explain why you
About we were talking about energy binding sites.
what about it?
Like, I can't remember much about IgG. It's like IgG has the power shape thing, right? If I remember the Y shape.
Yeah,
Yeah,
you go with the
but then
spaghetti.
hemoglobin has a prosthetic group. So the binding, it's not antigen binding. The binding site is in the sense that IgG is more antigen binding, but hemoglobin has prosthetic groups to bind.
Isn't it a comparison of the binding sites? It's a comparison. But the question, the second half of the question is more of explaining why
Then,
IgG has played so much.
use the content of similarities and differences and then explain diversity. Like, we can talk about how hemoglobin can technically only bind to protein C, but then IgG can bind to different kinds of antigens.
That is stating the difference that they have different diversity, but you're not explaining why they have different. You're telling me that they have different diversity, they bind differently, but you're not telling me why is that so. In the why, right, the diversity part, we will cover a lot more in page 2, but for now, it's basically your DNA.
Yeah, so we,
Right.
you mentioned the nucleic acid.
Yeah, so you need to know the genes that code for hemoglobin, you need to know the genes that code for IgG. And you need to know how variation can arise for IgG, which you will learn soon. But anyway, it is the somatic recombination. Somatic hypermutation. Task switching. Okay, you will learn this soon in H2 bio, these three processes will generate diversity for immunoglobulin in general. It's not just IgG, but immunoglobulin in general. Oh, sorry, you need to remove this.
Why?
Okay, so because the question is only on IgG, so it's already that specific class. So you cannot talk about class switching, so this is not relevant. So it's only these two. Somatic recombination and somatic hypermutation that can have the diversity, but hemoglobin doesn't have. There's only two genes, the alpha chain and the beta chain gene, that will code for the protein, and it must always combine that way. Okay, so that will be your topic areas for this question. Content, leave it to another time, or you can revise the content.
It's 6, right? 6 to 5.
Yeah, that's 6 is it.
It's like 5. That's 6. Unless the last two counts.
Oh, I can think of another one. But it's related to diversity. Somatic recombination is the VDJ recombination. If you don't know what is VDJ recombination. But besides that recombination, there is also the alpha chain and lambda chain. Never mind, okay, you don't learn so H2. But basically, under diversity, you can have, you should be able to have 3 calories. Okay, that's the graph. I hope that helps you a little bit. Okay, so whenever you encounter a question, the first thing that you should be thinking about is, after you have analyzed the question, know what is the key topic that the question is asking, then brainstorm for the different topic areas. That will be your outline. But after you brainstorm already, you need to actually have an argumentation, which is the next part. Okay, because your rubric for argumentation is that you need to have a sustained, coherent argument and a conclusion that shows insight. So this means that you cannot just be regurgitating all the content. You cannot just be telling me, listing down all the rows of ions, all the rows of proteins. This is just a list. But that is not an argument. So for you to have an argument, it's basically having a strong thesis statement, which means that you need a statement that you want to prove, a conclusion that you want to support. Okay, so all your points should be proving that one idea. So you should be structuring it in a way that you start off with the thesis statement, that is the statement that you want to prove, and then the subsequent paragraphs are all supporting that thesis statement. Or if you have counter arguments, then you need to counter the counter argument. Okay, you want to GP stuff. I don't get GP. So how to have a strong thesis statement for bio? Sometimes when the question only asks for content, it is quite difficult to think of what kind of thesis statement. So these are some points that you can use as guidelines to help you with thinking of a good argument. So you can talk about structure to function, you can talk about talk about energy gradients, but you need to talk about the significance of these gradients. Or you can talk about homostasis, etc. So it shouldn't be just the content, but it should be a significance of that function or that ion or that protein in that context. So you need to bring in some insight into what you're writing. Okay, so you can choose a unified concept, or you can choose anything that you feel that is insightful. These are just some guiding points. You can write on your points. And this leads me to again the 4 questions. But now I want you to write a thesis statement. So you can go and do the same question that you all were assigned just now. Write a thesis statement. Then after that, I will ask the other group to evaluate your thesis statement. So you all will solve.
How are we supposed to actually evaluate? Because compared to, for example, in teaching, where everything is very segmented, but then for bio, it's like more fractions. So how do we evaluate?
For now, I would just say that the thesis But if you don't have facts, then if it's just facts itself, then there's nothing to argue lah.
Then do we have to put examples to like this statement? Or these statements are like overarching?
The teaching statements are overarching,
So,
right?
like, it's a bit, for example, like, when you talk about iron, we have mentioned the topics that we're talking about.
No need. You can use the topics to link them together, but it is not necessary. You can use some other thing that is very overarching. Okay, this one I'll give you a bit more time because it's the first time you're doing this. 15 minutes, then 15 minutes we can go through and then we can call it a day. Yes, 5. That's 8 to 7, 5. It was last time.
Somehow this should be for us.
The question is here.
Discussion about, just a discussion about.
Do you make screenshots of what you can see, like the instances where you can see? Then I will send it to us through CRPC. Yes. Tech person. Excuse, send me some screenshots. Feedback from students. How do you support that? Think beyond the question. Maybe ask the question why or how. That might help you. Why? Discuss why, right?
Doing some like skills, like maybe each cell would
Or whether the functions range from these different skills. Not that it serves functions of all skills. I don't think they care so much about arguing against. They should minimally argue for the thesis. After all, it's not GPA.
quadrilaterals. And these are like the
Stability, I don't know.
well, that's not.
You all know the stability of different systems and all.
The structure is the
Structure.
the function.
Oh wait, but there's ribozymes and enzymes. You cannot have structural oligomylates. Why would the question for proteins here? It's crazy. No. But still, your enzymes, like, for your, like, each of these enzymes. You still need proteins.
But if you want to talk about RNU horror,
She's distracting me, bro. She's just too noisy,
so that's another part.
Ferrous ions are only
Yo, this video is not seeing you all. She got shh, bro. Don't put mute on.
Going to contact everyone.
ferrous ions are only
Okay, let's think about this. What is this statement? This is statement. Because we're not
we're not
we're not like it here.
We're not doing the contact. Because I have so much fun in the meeting out.
Yeah, I thought the flexibility in one, yeah, attendance, yeah. DTF is my name, bro.
Yeah, it's what thesis statement. Why so long for what? I only asked for thesis statement.
See, you not ATQ, you fail already.
Okay, while we are waiting, you can look at their question and comment on this long thing. Then you'll be faster finish your last question.
Flexibility. Our key word is flexibility. I think flexibility is our key word. So everything on this
Yeah, I should have been answered the very structure. But I think the main idea is this balance concentration is but it's really also. Okay, 5 minutes your comment is rather than That doesn't matter. Doesn't matter. You can just use all H2 content actually. Doesn't really matter.
It's giving the intro to the history.
Oh, that's how would it sound like for history or
I
something?
don't know. I feel like
At the level of history.
it's something like these oh yeah,
so that they can branch them into different. They're not like arguing the different roles. They're not really targeting the
Okay,
diversity.
the argument that they are trying to have is this one.
Yeah, but it doesn't really target like maybe their limitations.
So what's the better argument?
I would say ranging from different individual cell organelles to entire body system, they can add something like some proteins function better when acting alone, but some function better when acting in a system, like in the cells in the system, like kinds of systems. Then you're talking about diversity of space as well.
I mean, it's just a different perspective. You can write that as well. But I think this is fine too. I have a different comment. Yeah, you finish that. It's very strict. It's very strict. I need to match up to her level.
No, it's okay. Don't worry. Just for her.
Yeah.
Just for her.
For us, you can match down to our level.
No,
They give 10 out of 10 for all of it.
I need to match up to you. Their original thesis was actually protein serve a diverse roles in living things, which is literally this, but without this. That was the original.
Wait, after the second part was
Yeah.
Yeah. So the front part here about the wide variety of functions actually is not necessary, because that's what the question is asking already, to discuss the diversity of roles of proteins. So telling me that they serve a wide variety of function is repeating the question. But more importantly, I think the second half of the statement here is what you really need to support in your thesis statement.
Actually, a thesis statement can just be general. It doesn't have to include like evaluative criteria. Like also for GP, right? Thesis statements need to have some nuance.
Yeah, you just not
Can you tell me what evaluative criteria you can add here?
I don't
This
like.
one general paper or history bio?
Give me an evaluative criteria for diversity of roles of proteins.
Compare across context. Whether a person is healthy or I don't know.
Healthy or unhealthy?
Or like
Serious ah?
dying.
So if I'm dying, my this teenager more lah.
But that's because of mutations, right? That's not mutation.
She might not one that's already highlighted under evaluative.
Yeah, so to me, having this huge range,
Cell, like organelle to the various, I thought that can double up.
she sees it as to just a range of functions and not an evaluative criteria. That's what she's saying.
I think it's the
So I say I need to match up to the level where I'm talking about people.
No.
Oh, yeah, just
Yeah.
just her paper, right? Her paper,
So
right?
we need to match down to our level to match our paper.
Yeah.
No, I'm not going to match down, but I will match up to us for hers.
And actually, when you say compare across biological spaces, that's what we're referring to.
Yeah, I
The
think
cell
that's-
organelles to this. I'm not sure whether there's a need to talk about limitations with regard to the question rule.
Yeah, there isn't.
Yeah, so then in that case, it's not so relevant. But if you talk about the idea of comparing across biological spaces, which is a nice fanciful word for
Yeah, so this is the biological spaces.
There's different scales. I think that's evaluation.
Yeah. Okay, next question is ions. Okay, what are your comments?
Yeah. Oh my gosh.
Kind of makes sense. Not
Wait,
sure
I
why
don't know,
these
wait,
are very structure. I also don't know.
Wait. It's not like variation. It's like different kinds of ions. Yeah, that's what
I
it
don't
is.
know. Free chance.
No. Different kinds.
I agree with
So
that.
there's different ions and they are important because
Okay, if it's different ions, then the different ions are maybe like this statement on the basis. But like the other
on the balance of concentrations, but it also means that all your statements below need to be about that.
So why can't it be like you had a counter argument?
You can have a counter argument as well. You can have a counter.
Like so the one I wrote below is okay. Like if I just focus 3 body paragraphs on talking about the just answering the question and then one counter argument to for the balance and concentration.
Can you still need an overall conclusion? Sure. Okay. So just now I was reading. Where is the point? Did you change the point? Oh, I don't see the point. Oh yeah, I see that. Sorry. Cofactors. These cofactors you don't really need a balance in concentration, right?
Yeah.
Yeah. So this is not really a point to support. So the points to support the need for maintaining certain concentrations
It's like
is more for diffusion, regulation of concentrations in the ionic concentrations and therefore the osmotic pressure in the cell. So you can talk about that. You can talk about action potential. You can talk about the ATP synthesis, the proton gradient as well. Okay. But those ions that have individual roles, actually even sugar ions are, if you want to consider this as a pool of ions and then there is a balance so that you can serve as buffer, that's also possible.
the one I write the counter argument. Rows of ions in regulating gradient are only advantageous.
Up to you lah. But you are adding in a lot of additional criteria to your evaluation, which yes is important for GP but not so much for
In the top, right, because if you think along your line of biological spaces, your ions essentially in the categories that you put are examples when they act singly. Then some is the movement of ions that brings about a function. Some is the establishment of a difference like a mass versus a single ion, right? So I don't know whether that's where
that can actually be another way of writing the thesis statement as well. So you don't need to limit your thesis statement to just one function. You can have those overarching function to show that there is a different scale of function, but now the scale is not in terms of individual cells to the whole body system, but more of individual ions to having a pool of ions that makes up a gradient, and then a movement of ions across different spaces versus just concentrating in one area. So those are also different functions that you can talk about. Yeah. So for history bio, I think the counter argument is not so important. But what's more important is whatever statement that you have, you need to have evidence to support it. You just need to have evidence to support it. Sorry?
So it can be just 2 paragraphs, like 2 species.
Why do you have to do this?
actually it's the broad one.
Broad one.
Is then your sub-paragraphs is the first one that you
Yeah,
focus
topic
on.
sentences, yeah. Okay, that's the ions. Then for comparison of ribozymes and enzymes, let's factor more with what is. I realize that they don't like your.
It's quite good already. No, it's good already. It's good already. No, it's the second one.
Second one, oops. What about the second one? Less stable structure limited monomers might limit your points. That's true, it might limit your points. So how do you make it a bit broader but yet not too broad? Structural limitations. So you categorize this whole thing as just structural limitations that puts the constraints on the functions limiting the number of types of functions that they have, or the number of reactions that they can catalyze. That's possible. Okay. And the last one. Makes sense. Okay, well, IgG and hemoglobin share similarities in monomerase structure and binding sites. IgG possesses a higher flexibility. What do you mean by flexibility here?
Like flexibility to combine with more like some oil.
Then you need to be a bit more specific when you say flexibility, because I don't understand whether this flexibility refers to the structure itself is very flexible, you can move around, the arms can move around, or do you mean the shape of the binding site is a lot more varying, therefore more flexible to bind different things?
This is the question I got.
Compare the quaternary structure and binding sites. Actually, I thought that this question is easier to write a thesis statement because of the second half of the question.
Because it gives it to the stand already.
Yeah, so use that as the thesis statement.
Or pull out.
Yeah, so you don't need to write something else about higher flexibility. Or rather, this is where I don't understand what it means by higher flexibility, because you didn't explain more about what, in what context is it flexible. First part, a bit redundant. That's true. That's true. That's true. It's just that sometimes it's a bit abrupt if you just start from the second part. So, if it's a short part that is not really necessary, it's fine. But if you just need it to link things up, it's fine. Okay, so I hope this activity today helps you to better understand how to write an essay. Then the next lesson, I'll give you an essay to write. It's open book.
Maybe what topic?
It will be across topics from everything that you have learned so far, including H2, and H3. So core idea 1 to 3 like for H3.
We need to write a paper or we need to write a paper?
We try.
I'm going to try.
SLS.
Type. It's easier for me to mark also. I rather see type than handwritten words. You can write.
And look at the time based on SLS.
It will be time 45 minutes, including the planning of the question. So today you have to quite long
Can you
to
show
show
the two last question again?
right?
Because I think the thesis statement of these two questions or these two pattern of question might be very similar.
This one more on diversity, or that's what you mean. Because the enzymes have diverse roles, right?
True. Because the binding site of the IgG, the nature of the binding site contributes to the diversity. Then your same idea that was mentioned, right? Somebody was mentioning about the monomers. The fact is your enzymatic active site can be contributed by more monomeric.
A bit different because this is protein, all the monomers are and all amino acids.
But I got 20 mark.
Yeah.
And then in this context, hemoglobin, the binding part, right, is actually, although the amino acids contribute a bit of it, the bigger portion is by the FE. It's just whoever that said that comment about ribozyme, we talk about 4 monomers in nucleic acid bases.
Yeah. So that one you cannot use that one. Yeah, okay. So I shall put it on SLS. I will just add one more activity here. Then those who want to type it, you can type it. Those who want to write it, you can write it. Okay. It will be for the time.